Welcome to the deep dive. Today, we're taking a deep breath and plunging into the world of Rett syndrome. It's a truly devastating genetic disorder, primarily affecting young girls. We're talking about 1 in 10,000,
Yes.
roughly.
Yeah, that's about right.
And it's caused by mutations in a specific gene, MENCP2. But the real challenge here, as I understand it, is that it doesn't just hit one part of the body.
That's exactly right. You often hear it described as a neurological disorder, but honestly, Rett syndrome is just so much more complex than that. It's like a system-wide malfunction, really.
System-wide.
Yeah. I mean, beyond affecting speech, causing censures, and those, characteristic repetitive hand movements people might know about.
Right.
It also impacts the digestive system, muscles, bones, even the immune system.
Oh.
So trying to find one single treatment that can address all these different facets at the same time, that's been, well, the holy grail for a very long time.
Absolutely. And that's precisely what we want to unpack today. There's been a, truly groundbreaking discovery from a team at the Wyss Institute at Harvard University. They've combined an AI-driven drug discovery approach with some really innovative disease modeling.
Yeah, it's fascinating stuff.
And uncovered a promising new treatment for Rett. So we're going to explore how they actually did it, what this could mean for patients, right now, and also some pretty surprising new insights this work revealed about the disease itself. So are you ready?
Let's do it.
Let's dive in. Okay, so as you just touched upon, Rett syndrome isn't just about the brain. It's this profound challenge because it hits so many different organ systems.
Exactly.
To put it simply, it's like trying to fix a house that has problems with the plumbing and the electricity and the foundation all at once. You can't just send in a single plumber, right?
So absolutely not. You need a much more holistic approach. And that's what's made finding a single treatment so incredibly difficult for so long.
So what's the root cause again, that gene?
Right. The core issue is with the MECP2 gene, which is located on the X chromosome. And this mutation, it doesn't just cause one single isolated problem. It leads to a whole spectrum of impairments.
A spectrum, okay.
Think of it like maybe a faulty conductor leading an orchestra. Instead of just one instrument being out of tune, the whole performance gets affected, but in different ways in different sections.
That's a good analogy.
So the persistent challenge has always been, how do you develop a single therapy that can somehow retune that entire orchestra across all the body's very diverse tissues?
Okay, so given that, that's a pretty daunting challenge. How did this Wyss Institute team even start to tackle it? Especially when, traditional methods seem to have hit a wall.
Well, this is where their approach gets really interesting. The remarkable thing is just how different it was.
Okay.
They didn't go looking for, a single faulty on-off switch in a cell, the way traditional drug discovery often does. Instead, they developed this computational pipeline, an AI engine they call NemoCAD.
NemoCAD. Got it.
And this engine is what they call target agnostic,
Right.
meaning it doesn't search for one specific molecular target, like a single protein or enzyme. It looks at the entire gene network across multiple organ systems. It analyzes how the whole system, the whole network is, out of whack.
So it's like zooming out.
Exactly. Imagine it as this incredibly sophisticated diagnostic tool. Instead of just looking for one broken traffic light, it maps every single road in a city to see where all the different traffic jams are forming.
That makes sense.
And this AI, it actually grew out of a DARPA project, the Thor project, and it's already shown itself to be incredibly fast and effective in other areas.
That's a huge shift in thinking then. So this AI is looking at the whole system, the big picture. But it needed data, right? Real-world data to learn from, not just theory.
Precisely. You need a good model. And to accurately model the disease's multi-system impact, the team turned to something perhaps a little unexpected. Xenopus laevis tadpoles.
Frog tadpoles.
Yep, frog tadpoles. Using CRISPR gene editing technology, which is incredibly precise, they engineered mutations in the MECP2 gene in these tadpoles.
Okay.
And this let them create a really diverse model, one that reflected the variations you actually see in human patients.
And did it work? Did the tadpoles show symptoms?
They did, remarkably so. What's compelling is that these tiny tadpoles started showing key features of Rett syndrome.
Yeah.
Things like developmental and behavioral delays, seizures,
Wow.
and even measurable issues with their intestines, their muscles, and their brains. So this model was absolutely crucial. It allowed the researchers to analyze those gene expression changes across all these different affected organs, giving the AI that holistic, system-wide picture it needed to work with.
Okay, so you've got the AI engine, NemoCAD. You've got the data from these engineered tadpoles, comparing the gene expression in the sick ones versus the healthy ones.
Right.
So its mission then became predict which existing drugs could essentially rewire those faulty gene networks back towards normal.
That's the goal.
And it did this by searching through, what was it, the NIH's LINCS database?
That's right, LINCS. It holds gene expression signatures from almost 20,000 different drug compounds.
20,000. That's like asking a super smart librarian to find the one perfect book out of an entire massive library.
Exactly, a huge computational task. And the librarian, or rather NemoCAD, delivered. Vorinostat scored the highest on that list.
Vorinostat. Good.
It showed the strongest therapeutic effects in those engineered tadpoles. And the reversal of the disease features was genuinely impressive.
Like what specifically?
Well, it suppressed seizures. It corrected those unusual swimming motions, which seemed to resemble some of the motor behaviors seen in patients. And it even improved the gastrointestinal and muscular symptoms they observed in the tadpoles.
That sounds really promising.
And what's even more striking, perhaps, is that vorinostat was found to be much more effective, that's the quote, much more effective at suppressing these symptoms than trofinetide.
Trofinetide. That's the only drug currently approved for Rett, right?
That's the one. So finding something potentially more effective in these models is a pretty big deal.
Absolutely. But, you know, we always hear this caveat, what works in tadpoles, or even simpler models, doesn't always translate perfectly to mammals, let alone humans.
Yeah.
How did vorinostat hold up when they moved to more complex models?
That's always the crucial next step, isn't it? And vorinostat continued to impress. They tested it in preclinical mouse models of Rett syndrome.
Yeah.
It consistently reversed multiple symptoms. And here's a really significant point. It worked even when they administered it after the symptoms were already in full progression.
Oh, wow. So not just preventing symptoms, but reversing existing ones.
Exactly. Which is a huge distinction because trofinetide, the approved drug, wasn't able to achieve that same kind of reversal once symptoms had fully developed in those models.
That's important context.
It really is. And just for more context, the FDA had previously considered success in this specific type of late-stage mouse model a critical milestone for trofinetide's own approval pathway.
So it passed a key benchmark that the existing drug didn't in these preclinical tests.
In these specific tests, yes. And another promising sign, they found that an oral formulation of vorinostat also produced significant therapeutic outcomes in the mice. Easier delivery is always a plus.
Okay, so this is where it starts to get really exciting from a patient perspective, right?
Yeah.
Because vorinostat, it's already FDA approved for something else, a type of blood cancer, I think.
That's correct. It's approved for cutaneous T-cell lymphoma.
So that means Unravel Biosciences, the startup founded by these researchers, right?
Mm-hmm.
They can potentially rapidly repurpose it for Rett syndrome, cutting out years of early-stage drug development.
Precisely. That existing approval drastically, potentially, accelerates the timeline to get it to patients. It's a huge advantage.
So what's the status now?
Well, Unravel Biosciences' lead asset, which they call RVL-0001, that's their proprietary formulation of vorinostat, has already received orphan drug designation from the FDA.
Which helps speed things up for rare diseases.
Exactly. It provides incentives and streamlines the process. And they're gearing up for a proof-of-concept clinical trial set to begin later this year in Colombia.
Okay. How many patients? What's the plan?
It'll involve 15 female patients. And what's particularly innovative about this trial is the design they're using. It's called an N-of-1 trial design.
N of 1. What does that mean?
So instead of the usual approach giving one drug to a large group and looking at the average response across everyone,
Right.
an N-of-1 trial meticulously monitors how each individual patient responds to different treatments or maybe different dosages over time.
Ah, so it's like a personalized mini trial for every single participant.
That's a great way to put it. For a rare, complex disease like Rett, where every patient can present quite uniquely, this approach is seen as potentially much more powerful. It allows for really tailored insights and could potentially show benefit even with a small number of participants.
That personalized approach, yeah, that sounds exactly like what you'd want for a condition this complex.
Yeah.
Okay, so beyond just finding a potential new treatment, which is obviously huge,
Mm-hmm.
what did this whole deep dive using the AI and the tadpole models actually reveal about Rett syndrome itself? Did it give us any new fundamental understanding of how the disease actually works?
It absolutely did, and it actually threw a pretty significant curveball into some of the traditional ways we thought about the disease's underlying mechanism.
Oh, oh,
Well, historically, the understanding was basically that the MENCP2 protein regulates gene expression primarily by binding to DNA and interacting with other proteins, notably things called histone deacetylases, or HDACs.
HECs. Okay, those are involved in controlling gene activity, right? Like dimmer switches.
Exactly, that's a perfect analogy. They affect how tightly DNA is wound up, which
Yeah.
influences how much or how little a gene gets expressed.
Mhm.
So the traditional view is kind of that MECP2 managed this specific dimmer switch system.
Okay, makes sense. But I'm guessing the new findings showed it's not quite that simple.
Not quite. That's the unexpected twist. Their really groundbreaking finding was this. While histones, those spools DNA winds around in the brain cells of the Rett syndrome models, were indeed underacetylated, meaning the dimmer was turned down too low, as expected.
Mhm.
Surprisingly, they were overacetylated in other affected tissues, like the gastrointestinal tract.
Wait, overacetylated. So the dimmer switch was turned up too high in the gut but too low in the brain.
Precisely. It's
Yeah.
like discovering that in one room of the house, the lights are way too dim, but in another room, they're blindingly bright, and it seems to stem from the same faulty main electrical issue.
Wow. Okay, that fundamentally changes how you think about a single drug targeting just HDACs, doesn't it? If the problem is the opposite in different tissues.
Exactly right. It's a massive shift. It really helps explain why a simple one-size-fits-all drug targeting, say, just HDAC inhibition might not work or could even cause problems in some tissues. And it completely validates why their AI needed to look at the whole system. And they also pinpointed another key player, a protein called alpha-tubulin.
Alpha-tubulin, what does that do?
Think of alpha-tubulin as a crucial building block for the cell's internal skeleton, the cytoskeleton. It's particularly important for forming tiny hair-like structures called cilia.
Cilia, like in the lungs or...
Exactly. They help cells move things along or sense their environment. Very important in places like the gut lining and airways, but also involved in brain function.
Okay, so what about alpha-tubulin in Rett?
They found that the acetylation of alpha-tubulin, again, that dimmer switch idea, was also dysregulated, but mirroring that same pattern. It was hypoacetylated, dimmer too low, in brain tissue,
Right?
but hyperacetylated, dimmer too high, in other tissues, like the GI tract. And importantly, this pattern directly correlated with functional problems and inflammation they observed in those tissues.
So another molecule showing opposite problems in different organs.
Yes, and what's truly remarkable here is that vorinostat, the drug identified by the AI, it was able to reverse this messed up alpha-tubulin acetylation pattern in both directions. It helped normalize it whether it was too high or too low.
Whoa, so it's not just hitting one target, like the HECs.
That's the strong implication. It strongly suggests vorinostat has targets beyond just the well-known HDAC family, and it tells us that the multi-organ problems in Rett syndrome are likely caused by far more complex interconnected mechanisms than we previously appreciated. We need to keep exploring those pathways.
What a truly monumental step forward this research represents then, both for potentially treating Rett syndrome patients and just for understanding the disease.
Mm.
It really underscores the incredible power you get when you combine sophisticated AI with these really innovative biological models.
Absolutely.
And the speed you mentioned earlier, from the in silico prediction, on the computer, to the in vivo validation in living organisms within weeks. That's just astonishing.
It really collapses the timeline compared to traditional methods. And this whole approach, it isn't just a potential win for Rett syndrome.
No, I guess not.
This whole target agnostic strategy, looking at the network, plus the technology translation model they've set up with the startup, it really feels like a blueprint, doesn't it?
Yeah.
It could potentially be applied to tackle so many other rare, complex diseases that currently have few or no effective treatments. Places where traditional drug discovery has just, struggled to find a foothold.
It really makes you wonder then, what other complex multi-system diseases, the ones we currently consider maybe untreatable or just too complicated, what else might be on the cusp of a similar breakthrough if we keep embracing these kinds of AI-driven insights? Insights that really challenge our maybe older, more siloed understandings of how disease actually works.
It's a very exciting time for that kind of discovery. The potential is huge.
Truly fascinating. Well, that's all the time we have for this deep dive. Thank you so much for walking us through that.
My pleasure.